Hey everybody! Now that we've defined the three domains of life, bacteria, archaea, and eukarya, let's discuss what it means to be a prokaryote. Two of the domains of life are considered to be prokaryotic because they lack a membrane-bound nucleus. These domains are bacteria and archaea. The word prokaryote actually means pre-nucleus, and so just by the name itself, we can start to define what is a prokaryote. Let's draw two cells. The one on the left is a prokaryote. The one on the right is a eukaryote. You can tell that the cell on the right is a eukaryote because it has a membrane-bound nucleus. The nucleus is important because it contains the genetic code of the organism. So, what does this mean for the prokaryote? It must have its own genetic material too. Even though its genetic material is not enclosed by a nuclear membrane, it is still concentrated in the part of the cell that we call the nucleoid. Let's add some scale bars to show the size of these prokaryotic and eukaryotic cells. You can see that the prokaryotic cell is much smaller. Because it is much smaller, the prokaryotic cell can more easily meet its nutritional needs and tends to reproduce much more quickly. The prokaryotic cell is also much simpler. It doesn't have a nucleus, and the prokaryotic genome is less complicated. Most prokaryotes contain a single circular chromosome. Humans have 23 pairs of chromosomes. This makes for over 3 billion base pairs of genetic code versus approximately 4.6 million base pairs for a bacterium such as E. coli. Whether it's 4.6 million or 3 billion base pairs, that's a lot of information to pack into a single tiny cell. How do cells do it? Well, for eukaryotic cells, there are proteins called histones that the DNA wraps around. Each bundle of DNA and histone is called a nucleosome. The nucleosomes and subsequent coiling of the DNA allows for the DNA to be compact and appear like this right before the eukaryotic cell is going to divide. Prokaryotic cells don't have histones. They do have nucleoid-associated proteins that also bind to DNA, but do not form defined compact structures like nucleosomes. So prokaryotes primarily rely upon supercoiling and looping of DNA to compact their DNA. Besides compacting DNA, supercoiling plays an important role in whether or not a particular gene or segment of genetic info is going to be read and used to make a new protein. So, the degree of compactness for the DNA is going to vary depending upon the cell's growth conditions and its response to environmental cues. The accumulation of supercoils can act as a topological barrier or physically preventing certain segments of the DNA from being expressed at a given time. In this way, cells can be efficient by only expressing the parts of the genetic code to make more cellular components when those components are needed. Eukaryotes have a nucleus, and so they've physically separated the processes of reading the genetic material and making proteins. The flow of how genetic material flows from a DNA sequence to a protein product is often called the central dogma of molecular cell biology. What happens in the cell is that DNA is transcribed to RNA. In turn, the RNA is translated to protein. Both processes are essential because our cells don't make proteins directly from DNA. To make proteins, special organelles in the cell, called ribosomes, need to have the information in a way that they can decode. Ribosomes cannot read DNA, but they can read RNA. Just imagine how each part of the cell has a particular function, and each part of the cell requires a certain set of instructions presented in a language they can understand. RNA polymerase reads DNA and transcribes the parts that need to be expressed to make instructions in the form of RNA. This RNA is subsequently read by ribosomes to make a protein sequence. Since eukaryotes have a nucleus, the process of transcription occurs in the nucleus, and the process of translation occurs outside of the nucleus. Prokaryotes, which lack a nucleus, do not have a physical barrier separating these two processes, and so transcription and translation can occur at virtually the same time. We will wrap up this short video segment by talking about cell walls. Cell walls are external to the cell's plasma membrane and provide structural support in addition to helping a cell be more resistant to osmotic changes. Some eukaryotic cells have cell walls. For example, fungi, plants, and algae have cell walls. But animals like us humans do not have cell walls. Many prokaryotic cells do have cell walls. Bacteria have cell walls, and some archaea species also have cell walls. The chemical composition of these cell walls is another feature that distinguishes eukaryotes and prokaryotes. We can also use the properties of the cell walls in order to distinguish between specific bacteria. The Gram stain and acid-fast stain both make use of features of bacterial cell walls to distinguish between groups of bacteria.